I will move on to our next speaker. We actually have two speakers for this next one. To start us off will be Dr. Bhanu Telugu as a representative of the U.S. Department of Agriculture.
First of all, thank you, Dr. Kurtz, and I'd also like to thank leader NS To-Do and the program committee for extending the kind invitation to talk here today. I have to make a correction. I do not represent USDA. I represent University of Maryland. I incidentally have an appointment with the USDA, and my lab is located there. So the reason we have three speakers here is we wanted to bring a perspective from the standpoint of an investigator who is already performing genome editing in large animals, primarily pigs, on a federal facility, a veterinarian who's on the animal use and care and also on the local oversight committee, and finally, a former regulator who is actually can represent USDA, Dr. Diane Ray Cohen. So what we want to do is kind of bring about, briefly provide an introduction and overview as to why we are doing genome editing in agricultural animals. So from the biomedical standpoint, it has very well discussed why we are doing genome editing. We want to model the diseases and so forth. Why do we need to do genome editing in agricultural animals for agricultural applications? We would like to discuss the current regulatory paradigms that already exist in place. And finally, kind of provide a case study as to how genome editing animals are overseen at a federal facility in the United States. So it gives you a full circle of what's happening here. So most of the speakers behind us, we already talked about this. Genome editing or affecting change in the animal genome is not a novel concept. We're not doing this all of a sudden from 80s using transgenics or now with editing. We've been doing this for centuries. But it took different shapes. So we've been doing this as part of agriculture. So we've been using mass selection, or we've been using more refined processes like pedigree selection, marker-assisted selection. So the way we are selecting the animals have been changing, but we're still causing or inducing changes in the animals by a different tool called breeding. So with that being said, the transgenics came into play in the 80s. And it's been a game changer, and it still is for one reason, which is that we have now the ability to introduce novel traits into the animals, which do not exist otherwise. And secondly, it can introduce the changes much more effectively than the traditional breeding itself. Then with the advent of genomics, now we use genome-wide selection to comprehensively select for the traits. And then comes the topic of today's discussion, which is genome editing. So what we call is as a precision breeding or rational selection of traits. That's what I would like to call them. So why do we do genome editing in livestock? So I'd like to argue that there is a bottleneck for the current breeding. So Scott alluded to this in his talk previously. And some may argue, well, the breeding has served us tremendously well. Why do we need to do genome editing for agricultural animals? Well, there's a good example here. If you just take the case of a dairy animal, in the last 40 years, using just breeding alone, we have increased the output of the milk per an average cow in the United States by 100%. Remember, it's 100%. So we went all the way from 12,000 pounds to currently they produce about 24 to 26,000 pounds per animal. And this has been done all by breeding alone. So why? Well, if you look at the other parameters in these animals, so if you look at the reproductive performance as it is to a daughter pregnancy rate, you can see that during the same period, the reproductive performance of these animals have declined. Not to mention that now they're susceptible more to mastitis, metabolic disease, and so forth. So a singular focus on, or singular selection focus on a particular trait has resulted in negative consequences elsewhere in the genome. So there are several examples to it. If you take a poultry, for example, a selection or a drive to produce more meat has resulted in brittle bone or the bones disorders in these animals. So there are several such examples. We can spend a whole day on this. So what we also call it as a genetic bottleneck. The reason is that Scott has alluded how the genes are linked together that cannot be removed through breeding alone. So I'm going to provide a simple illustration, probably drives the point much better. So for example, I'm going to use two haplotypes. Remember, the haplotypes are the segments of DNA that gets dissociated during the meiosis process, right? So consider there are two haplotypes, haplotype A and haplotype B. And in haplotype A, you have a desired allele, and closely next to it, there was an undesired allele. So remember, during meiosis, they always break together. So they always are inherited as one unit. So every time you get a desired allele, you'll also have that undesired allele that tags along with it. And on haplotype B, consider that there is a polymorphism that is advantageous, and there's another one on a different allele, which is undesirable. So what would happen during meiosis? So you have 4 gametes, right? So you have 4 different combinations. So if you look at the favorable alleles only, this allele only, you only have 2 gametes that inherit that. And if you look at the desired QTN, which doesn't show very well, it only shows up in 2 of these gametes. And if you want to look at both of them, it's only inherited in 1 gamete. So what we are looking at here, we're looking at low heritability. So even though you may have a desired allele, the heritability of that allele in the next generation is very low. Not to mention that even in this gamete, you still are carrying that undesired allele. So what can we do? So this is where genome editing can be useful. So imagine a situation where Scott goes and knocks out that undesirable allele on that haplotype A. So now all of a sudden, and I go and change this QTN to a favorable QTN. So all of a sudden now we have a scenario wherein the heritability is much more higher for the QTNs. For example, if you take the levels of QTNs, all of them have the desirable QTN. And if you look at the desirable allele, at least 2 of them carry those. So this is what we call as directed selection or rational selection of traits. We can force these much higher. We can increase the heritability. We can improve the selection process of these animals. So when we talk about genome editing, we're talking about gene knockouts, I mentioned, so where we're deleting these alleles. It could be gene modifications. We're changing the amino acid sequences. And we're also talking, in some instances, the knock-in. So the polled allele is a very good example of a knock-in, right? And the rationale here is that, as I briefly mentioned, it improves, overcome otherwise low heritability of the traits. It separates the linked genes, and it increases the efficiency and precision of the desirable traits. So conventional breeding is random. This is more directed. That is the idea here. So, and not to mention that introduction of traits would now be facilitated by this use of the editors. So with introducing a breaks, now you can introgress these alleles at a much focused levels in the genome. So I'm going to provide a brief overview of what we are doing at USDA. So as far as genome editing, we are doing all 3 of them. We are doing the knockouts, we are doing the modifications, and we are doing the knock-ins. And we're doing all of them in the embryos directly. So if you look at this example, I don't have that dark embryo that you showed because it is really dark and doesn't show anything. So consider this illustration. So what we do is we go into the pig embryos and we inject the CRISPRs. So herein you inject the nuclease along with the guide RNA. So what happens? Well, this is what happens. In one generation, for example, from one pregnancy or one trial, we have 18 piglets, and all of them are edited. So as Scott was mentioning, even a 3-year-old can make CRISPRs. I'm not a 3-year-old, but I definitely jumped like a 3-year-old when I saw this result. I was exhilarated. For the first time in the world, I could make a knockout animal in 4 months. And not one of them, I made 18 of them. So this was a fascinating result for us. And by the way, these are the animals. They are no different from any of the animals that you see outside. And they're very happy too. So in terms of altering the SNPs or the QTNs, so this is an example where we wanted to change 2 amino acids that are in tandem, in close relation to one another. So again, we went into the embryos, we injected the CRISPRs along with the guide sequence. We changed these 2 nucleotides here, and we changed another 2 nucleotides here. So with the first one, we could screen this because when we introduce this change, we lose that AFL2 restriction enzyme site. So how does it look? So if you run on a gel, wild type would have these 2 fragments that are running closely that have the AFL2 site. When you do the editing, they lose those AFL2 site. Well, that's not convincing enough. We go and sequence them. And we show that we actually can change those sequences. So here an example where we actually changed the nucleotide sequence. And finally, introducing short stretches of sequences. Dr. Schneeke was talking about putting in LOXP sites. So we wanted to see if we can introduce small stretches of sequences. So again, in this case, again, going directly into the embryos, we injected the guide sequences alongside the targeting vectors to introduce this 34 nucleotide sequence called LOXP site. And as you can see here, like in the embryos, some of the embryos, you don't see any modifications. Some of them you see, well, it's showing up. Well, some of them you have the deletions. And in a whole bunch of them, you can see that 34 nucleotide shift in sequence, which is the introduction of the LOXP site. So we have demonstrated that you can do knockouts, knock-ins, and do the precise modifications by injecting the CRISPRs or the editors directly into the embryos without having to do any pre-screening, do anything with the cells, go directly into the embryos and affect the change. So the potential benefits of these technologies as I envision them, one, they facilitate precise modifications, and they are more faster, more reliable, and cheaper than conventional breeding. Just to put perspective in terms of, for example, if you're looking at a cattle project, he was talking about how, for example, to do 5 generations of breeding to introgress or homogenize a trait, you can literally spend 30 years doing just that in cattle. And if that includes site selection, by the way. So, but in this case, we can just go in 1 generation and cause the same exact change in 1 generation. So it could be 4 months in pigs and 1 year in case of cattle. What are the potential risks? So there are always going to be risks associated with some advantages. You have heard a whole lot of this off-target effects. Don't ask me what happens in my pigs with these off-targets because I have not looked at them so far. I'm just hoping for a day when somebody comes and says, these are the safest possible CRISPRs, use it, and you don't have any off-targets. I'm going to wait for that. But as of now, yes, there are some associated off-targets, but that's really not a focus at this point. The animal welfare has not been compromised with the use of editors. There could be a potential for reducing genetic diversity if you're making a founder herd and just populating the herd with them. You can end up with a inbreeding effect, so as to speak. You can be reducing the traits. And one word of caution is, like we said, all the dairy industry in the United States is driven by 8 bulls. Well, it could be very well in the future, it could be driven by only 2 polled cattle at recombinetics. So, no, okay. And finally, this does not pertain to all of the species, but there could be an issue of biocontainment. This is certainly not the case with pigs, not the case with cattle, but there can be situations like insects, moths, and other species in which this could be an issue. So these are the perceived potential risks. Any additional legal and ethical concerns with this? There could be perceived novel environmental risks. There are some advantages, by the way. A very good example is the EnviroPigs, so the ones that produce phosphatase. So there are cases where we can use them for the advantage. There could also be some risks associated with it. Misuse of this technology for entertainment purposes or like this one, potentially nefarious reasons. A good example is the micropigs that have been generated. They've been generated as pets. But I'm going to ask you guys, how is that different from designer dog breeding? So everything we need to put it in a context. So I'm going to leave it there. And application of this technology to human germline editing. That is a potential risk that needs to be taken seriously. I'm not mentioning here, somatic editing is acceptable. What we are worried about is human germline editing. So how do we regulate genome-edited animals? So for the first time in the 80s, we went into this scenario when the transgenic animals first came into foray. So we went from a period of certainty to all of a sudden we thought, oh, okay, God, we got recombinant DNA animals, so now we are at risk. So anytime we think about something novel that we don't understand, we go into this panic. So there is uncertainty. So out of this, we actually, there was an establishment of a regulatory oversight for the use of transgenic animals, and we call this a coordinated framework. So in the United States, there are 3 agencies. So you have the USDA, FDA and EPA, they regulate various aspects of the genetically modified animals. So from a USDA perspective, they are worried about the safety of the animal, the agriculture, and the environment. From the FDA perspective, they want to see if it is good for food and feed. And finally, from an EPA standpoint, they're interested in safety for environment. This is a very simplistic scenario. Actually, it's much more complicated than that. Ask Malini if she's around. So some of the general principles of this coordinated framework, I want you to emphasize, and I really want to drive this point, USDA, US Department of Agriculture, does not regulate GE animals. So FDA has emerged as the de facto enforcers of this technology. So the risks of genetically engineered animals are not fundamentally different from those that exist from non-genetically engineered animals. The existing laws provide adequate authority. I don't think just because we have genome editing means that we have to come up with new regulations. The ones that we already have in place do suffice. And Dr. Harper will mention how some of this can be maintained at a local level as well. And the regulation is always, we keep repeating that it's always science-based. It's always the product, not the process. So genome-edited animals would be regulated. So that being said, how are the edited animals are regulated genetically engineered animals are regulated in the United States? Like Malini mentioned very briefly, the regulator of genetically engineered animals, anything that contains a heritable DNA construct is considered a trigger, and that would be followed upon. The new animal approval is based on whether the animals are safe, effective, and for the intended use. So again, it's not the process. What do you use for the process? It does not really apply here, because I think in the end, it's always the product that's been regulated. So all is not gloom here. We have good examples of how the genetically modified animals are already making their way. There are very few examples. I mean, glowfish, I think we'll hear from Alan tomorrow. Atrian goat, where the production of these pharmaceutical products from the goat milk has already been approved. We have the oxytec mosquitoes. Enviropig is on hold, but it may come back. And hooray, we have the aquadvantage salmon that's been approved. So I'm hopeful that in the near future, this is going to pave the way for other edited animals. So with this, I'm going to hand over to my colleague who's going to talk about the local oversight processes for these edited animals in the United States, just to provide a perspective for you guys.
And as Dr. Telugu mentioned, my name is Susan Harper, and I am with USDA with the Agricultural Research Service, which is distinct from APHIS, which is the regulatory wing of USDA that provides oversight for animal welfare. So basically, when Dr. Telugu's project came to the ARS for evaluation, well, the first thing we wanted to do as a committee, the Animal Care and Use Committee, is we wanted to look at the provisions for the regulation of transgenic animals and understand that those did not necessarily apply to animals that are produced through genome editing technologies. There were regulations in place to protect the welfare of animals and the safety of workers, the safety of the environment, and also the public. And these oversight, local oversight entities were primarily concentrated in the Animal Care and Use Committee, the Institutional Animal Care and Use Committee, or IACUC, and the Institutional Biosafety Committee, or IBC. And we'll talk a little bit more about each of those committees. The Institutional Animal Care and Use Committee is required by the animal welfare regulations when regulated species are used for research, teaching, or testing. Regulated species do not include the traditional laboratory mice, rats, poultry, fish, or animals that are agricultural species that are used for food and fiber or agricultural research. Also, if you receive PHS funding from NIH, CDC, or FDA, then you're required to adhere to PHS policy, and PHS policy also requires the establishment of an Animal Care and Use Committee. There are 2 publications which guide the standards used in animal research. One is the National Research Council's Guide for the Care and Use of Laboratory Animals, which pertains to traditional laboratory animal species, and the other is the Federation of Animal Societies Agricultural Guide for Agricultural Species, which pertains to agricultural research. Both of these publications also require the establishment of an Animal Care and Use Committee for oversight of animal research. The emphasis that the Animal Care and Use Committee follows is they're looking after the welfare of the animals, the safety and the welfare of the animals. And these are animals that are used for research, teaching, or testing. The Animal Care and Use Committee typically will conduct a harm-benefit analysis to ensure that the use of animals is justified and appropriate for the application that's being proposed. Typically, the 2 guides do address genetic research that involves animals, but the Animal Welfare Act regulations and PHS policy do not specifically address genetic technologies. USDA APHIS has passed Animal Care Policy number 10, which specifies that any facilities that produce cloned animals for interstate commerce, regulated purposes, have to be licensed, and also that any facilities that produce novel, genetically engineered animals must be registered as research facilities. So that did sort of pull these types of programs in under the Animal Welfare Act regulations. Of significance to the research that we're talking about right now is that it was conducted at a federal facility and it did involve agricultural applications. So the Animal Welfare Act regulations did not apply to this field of research. In addition to the Animal Care and Use Committee, there's also a requirement for an institutional biosafety committee if your institution receives funding from NIH. This is through the NIH guidelines for recombinant and synthetic nucleic acid molecules, and it does have provisions for the use of transgenic animals, the production or use of transgenic animals in research. There are specific requirements when you're producing transgenic animals. If the animals are large enough, they have to be identified within 72 hours of birth, and there have to be provisions for the disposal of the animals at the end of the study. And because there's a potential for accidental release into the environment, there has to be distinct and biochemically assayable DNA sequences so that you can identify those animals and from the non-transgenic population. In addition, the NIH guidelines include a lot of information on biosafety and biocontainment practices that you have to follow to do this type of research. In addition to those 2 primary oversight entities, a number of other miscellaneous regulations sometimes apply when agricultural research is involved. Number 1 is the Clean Water Act, and this primarily pertains to any kind of wastewater runoff from facilities where genetically engineered animals are housed or maintained. OSHA regulations pertain to the safety of workers who are involved in this type of research. And there's a publication from CDC NIH called Biosafety in Microbiological and Biomedical Laboratories, shorthand is BMBL. This includes laboratory safety requirements for any kind of research that involves biohazards. And if you are doing any type of research with the select agents or in toxins, you're required to, the way your program will be evaluated is based on the information in the BMBL. In addition to these general documents, states and different municipalities may have local regulations that pertain to environmental protection or worker safety. And there are also always evolving international standards that try to harmonize the regulations between different countries for any kind of research that involves genetically engineered animals. So to recap, the research project that we reviewed that Dr. Telugu has submitted, the goal of this project was to produce a line of pigs that would not serve as a reservoir of influenza. The first part of the project was to create the pigs and then create a foundation line with offspring. And then with the offspring, influenza challenges would be conducted to test the integrity of the line. In addition to testing the animal's performance, we were also monitoring the animals for adverse genetic and phenotypic stability, as well as the, any unintended consequences that may have resulted from the genetic editing. The first thing that we asked was, do we have appropriate local oversight mechanisms in place to cover this type of research? And for USDA ARS programs, they're required to have an animal care and use committee if they do conduct any kind of research involving live animals. And they're also required to have an institutional biosafety committee if they're conducting any research that involves any kind of recombinant or synthetic nucleic acid molecules or any type of biohazards. So basically, this study involved 3 different institutions, and the project was reviewed by the IACUC and the IBC at all 3 institutions. One was university, and there were 2 ARS locations. The review process that we followed was the basic review process that we would follow for any other project. We looked at animal welfare implications, biosecurity concerns, laboratory safety, potential occupational health issues, and containment and confinement practices. We also made sure that there were agreements in place between the different institutions to define oversight responsibilities, animal ownership, and any other issues that may come up during the course of the study. Based on this initial review, we have an N of 1 so far. This is the first study that we reviewed this, of this type. We have not seen any gaps in the local oversight system as a result of the Institutional Biosafety Committee and the Institutional Animal Care and Use Committee's oversight of this research.
So with this, I'd like to thank the members in my lab who performed this work and the funding sources. What we really wanted to show here is that, irrespective of the regulations that may or may not exist, there is adequate oversight on this kind of work, especially genome editing, that ensures the welfare of the animals, the health workers, the safety of the workers, and so forth. There is a common perception, at least in some people, that animal agriculturalists are a bunch of cowboys who do whatever they want. That is not the case. I really want to emphasize that. And another thing we would like to drive here is that there are adequate measures in place, and that we really don't need to come up with new regulations. There are already enough regulations to cover what we are doing. All right, thank you very much.